today I'm super excited to be speaking with Professor Bryan Roth, who's the Michael Hooker Distinguished Professor of Protein Therapeutics and Translational Proteomics at UNC School of Medicine at Chapel Hill, and very recently, as of the last few days, an advisor to Xylo Bio. So great to be speaking with you, Bryan.
Great to speak with you.
Yeah. Obviously, your lab's made countless sort of contributions to some very fundamental science and translationally relevant science in the field of Class A GPCRs broadly. How long has your lab been interested in psychedelics for? I imagine it goes back quite some time.
Yeah, since the 80s.
Nice. Yeah.
A
Yeah.
long time,
Yeah. But
basically. Yeah.
before it was cool, you know?
Yeah. I actually first started studying 5-HT2, what we now know as 5-HT2A receptors. They were then called 5-HT2. And there was a debate in the field about whether the 5-HT1 receptor or the 5-HT2 receptor was the real serotonin receptor.
Right.
Okay. And at that time, there was very little known about the 5-HT2 receptor, now the 2A. And the person that I was working with, this very famous neuropharmacologist, Mimo Costa at the time, was the greatest neuropharmacologist in the world. He had this idea, this very bizarre idea, that serotonin was not the ligand for the 5-HT2 receptor, that he believed there was a peptide, because serotonin has pretty low affinity for that receptor. And there were some unusual things about how the receptor was regulated, which I'm not going to go into here. But so my task was to find the peptide ligand for the 5-HT2 receptor. Okay. And after 2 years in the cold room, I told him it's serotonin.
Yeah. The peptide hunt was unsuccessful, I'm going to guess.
Yes.
Yeah, this is an excellent point as we sort of understand. Obviously, the 5-HT family of receptors is now quite well described, and I'm familiar with some of that history. You know, Richard Glennon was one of my thesis examiners. He was also doing a lot of work at the very beginning. Once relayed to me a story very early on in the field when he was presenting some of his early work on 5-HT at a conference with a poster, and had some other academics sort of look at it, sort of dismiss it and keep walking, and you know, mentioning how it was sort of, you know, this irrelevant curiosity, which we obviously now know is not quite true. So your latest paper, on which Manesh is the first author, is a pretty impressive body of work, because it's that, you know, there's a lot of interest in which targets are relevant, therapeutically relevant for psychedelics. And as far as I know, this is definitely the largest screen at the receptor and signal transduction level of the largest collection of psychedelics and sort of related compounds. Yeah. So are you able to describe just briefly, whoever's listening, the scope of that paper?
Yeah, so the idea was, because there's no reliable information on the sort of broad pharmacology of psychedelics. And we were fortunate to get a very large grant from DARPA about 5 years ago, which provided basically the resources so that we could begin to address these, you know, really fundamental questions related to psychedelic drug action. So in addition to, you know, a lot of drug discovery and structural biology, we also, you know, basically evaluated the pharmacology of LSD essentially across the druggable genome. So we actually screened LSD against the kinome and the GPCRome. And LSD actually has some, you know, it has sort of weak activity at some kinases, but we focused on both kinases and GPCRs because those two families comprise probably, you know, 60% of the druggable genome, what is currently thought to be the druggable genome. And so we thought we would get a pretty unbiased perspective of the pharmacology, both the on and off target pharmacology of psychedelics. So we initially looked at LSD. You know, of course, LSD is extremely promiscuous. And then I got together with Dave Nichols, who of course is, you know, very well known in the field of psychedelic research. And he and folks in my lab were basically put together a list of 41 psychedelics from all the chemical classes.
Yeah, I like that there was a really representative selection of tryptamines, phenethylamines, lysergamides, and even some of these sort of adjacent compounds that are not so widely studied. Yeah, exactly.
MDMA, MDA, methylone, glyceride. MK212.
Yep.
So a lot of those other interesting compounds were evaluated as well. And, perhaps not surprising, most of the psychedelics, at least the tryptamine and the indole-derived psychedelics or ergoline-derived psychedelics interact with essentially all serotonin receptors. Mm-hmm. They're pretty potent and efficacious agonists. There are, interesting differences within the drugs, but by and large, they all have, extremely promiscuous pharmacologies. And many of them actually are agonists at dopamine receptors. So one of the sort of big surprises we found was that psilocybin, psilocin, actually is a pretty good D1 agonist.
Has that been reported previously?
That was never reported before.
Yeah, I think a lot of work with lysergamides, right? Their dopaminergic activity is quite well described, but yeah, I was a little shocked to see that.
Yeah. So that was something we discovered really early on. The other thing that was interesting is that the phenethylamines in general are more selective.
Mm-hmm. But for the 5-HT2 set of receptors, right?
5-HT2 family, yeah. They're generally not selective across the class, but they generally are more selective for the 5-HT2 family. They also hit 5-HT7 receptors, most of them, which is like really interesting, potentially really important. And the other big thing, so there's all this data basically, and I have a Substack where I've gone into some of this stuff already.
Yeah, I've been following the Substack. I like that it's frequently published.
Yeah, but I guess the two other big things were that all the psychedelics activate the 5-HT2B receptor, which, is potentially bad news. Mm-hmm. And that the report that psychedelics preferentially activate the TrkB receptor appears to be non-replicable.
Yeah, that was one of the more interesting parts at the end of the paper. When you look at the whole kinome, we received a lot of questions at the company about the relevance of our compounds and their engagement with TrkB, and I was somewhat dismissive of the finding initially until it was replicated by other labs. And I'm now aware that this paper has failed to replicate it. I believe Dalibor Samesh's lab at Columbia, although it's not published, he had a nice sort of Twitter thread describing a number of experiments they'd run to also sort of show that these compounds are not, psychedelics generally are not high affinity ligands for TrkB. So it's nice to see some refutation of that.
Yeah, David Olson's recent paper in Nature Neuroscience, buried in there, they also show that their compound and other compounds do not activate TrkB.
Right, yes.
Let me just say that when the paper came out, I actually was really excited about it. And I was skeptical because the chemistry didn't make any sense. When I looked at the binding site, it just, it didn't make any sense. But we thought that, it might be true. So I enlisted Brian Choiquette to start doing docking studies. And he immediately told me that their docking results are not up to par, basically.
Right.
I'll just put it that way.
Yep.
And so he was looking for other sites. And I said, well, so what we were going to do is we were going to purify TrkB and solve the structure with LSD and then use that for docking, right? So I had 3 postdocs beginning to purify the TrkB receptor. And Brian was, they were building models and everything. And I said, well, maybe what we should do is see if we can replicate these findings first.
Yeah. Yeah, getting a solved structure would definitely be a very elegant solution, but basic replication might be a faster path to justifying that effort.
Yeah, and some data that we didn't put in the paper, but we sort of mentioned it in the paper, was they did binding assays with radioactive LSD. And of course, my lab does, literally thousands of binding assays a day. So I run a screening center. So those experiments were like really easy for us to do. And there's absolutely no specific binding of LSD when you transfect cells with the full-length TrkB receptor under, it's not there. I mean, there may be something there, but it's just not anything we can replicate. So.
Mm-hmm. Were there any other big findings from the paper that were a little surprising to you in terms of potential therapeutic relevance for the class broadly?
Well, for DMT, there's been this, a lot of verbiage on the sigma receptor, that it's quote unquote a selective ligand for the sigma receptor. It turns out that is completely untrue.
So I don't know if we discussed this at GRC, but my PhD thesis was actually on polycarbocyclic amines targeting the sigma receptor, and
Okay.
I propose it as one of these targets. There are many almost every drug, practically in
If you have
psychiatry
a
hits
drug that's
sigma.
a drug, it will hit the sigma
Yeah,
receptor. Yes.
exactly. But there are no approved selective sigma ligands, as far as I'm aware, and the
Oh,
companies that were developing them historically, I haven't seen much progress there in the clinical trials for those agents. So I think it's a extremely promiscuous sort of target for a lot of simple aminergic compounds.
so we tested them all against the sigma 1 and the sigma 2, and that
Right.
data is in the paper. And virtually every psychedelic has the same affinity for the sigma receptor as DMT. They're all around a micromolar.
Yeah, so nothing magical about it.
About DMT and the sigma receptor.
Right, yeah. I've also heard that proposal that it's unique to DMT, and I remember thinking that you can pretty quickly demonstrate to yourself that's not true when you look at a number of other related substances. So what's your sort of view on the polypharmacological approach for psychedelics versus more selective approach at some of these targets, 2A or otherwise? It's pretty well accepted at this point that the history of psychiatric drug development has been polypharmacological, but it's also
Mhm.
been, largely driven by, serendipity, I would argue, historically in the sort of golden age of psychiatry. Do you think there's a place for a more selective approach in psychiatry generally with regard to target selection and target engagement?
Yeah. So for psychedelics, that is a really interesting question. I'll just give you my thoughts on it. Okay. I suspect that unopposed 2A agonism is not necessarily a good thing.
Right.
That's my guess. And this is based on reports with drugs like 25-cyano-NBO. So we actually have found that those compounds, of all the compounds we've looked at, are the most selective for the 5-HT2 family of receptors. And 25-cyano in our hands is the most selective one out there. And, there's some toxicity associated with these compounds, clearly.
Yeah, that's a good point. So there's two interesting points here. One is that those compounds are uniquely toxic relative to other psychedelics. It seems
Mhm.
like, psychedelics are notable for their sort of remarkable lack of acute toxicity.
Yeah,
And you
yeah.
look at the LD50 with an N of 1 in elephants and LSD historically to sort of demonstrate the point. So these are remarkably acutely safe compounds in terms of their toxicity profile. Some of the N-bombs and related compounds, these N-benzoyl phenethylamines, have been associated with sort of overdose type deaths in the
Hmm.
population with recreational use, but they're also sort of the only class of psychedelics that we know of that are selective in that way for sort of 2A, 2B, and 2C. They do seem to have very high levels of selectivity. So do you think this is maybe an issue related to the specific chemical scaffold or more generally to the sort of pharmacological profile of those drugs?
We don't know, obviously.
Yeah, yeah.
My suspicion, so 5-HT2 receptors are found throughout the body, platelets, blood vessels. 5-HT2A agonists promote platelet aggregation and blood vessel constriction. So my big concern has always been stroke
Right.
with these really potent 2A agonists that are highly selective. The other thing is that the off-target activity, particularly probably 5-HT1A, maybe some other 5-HT1 receptors, I think tends to moderate the experience because those basically inhibit pyramidal neuron firing, but they won't inhibit the biochemical effects of 5-HT2A agonists. So current understanding is that the plasticity and other things occur through sort of canonical GPCR signaling unrelated to the electrical activity. My suspicion is that some of these off-target effects might actually be beneficial and or potential therapeutic, but we don't know.
Yeah.
We don't have any of those compounds in humans. That's basically what we need to do to find
Right.
out.
Yeah, I think there's definitely something to be said for developing novel classes, selective or otherwise, and really getting to first in human studies as quickly as possible. So I appreciate on your recent Substack the idea that animal models are quite critical for the development of drugs in psychiatry, but ultimately no animal model alone or in combination can substitute for just a properly designed first in human study.
Yeah,
There's
particularly
no other way to
drugs
do it.
like, we don't have the foggiest idea what's going on in a mouse's head when we give the mouse LSD.
Yeah.
And, about the only thing we can probably predict reliably is whether the drug is going to be psychedelic.
Right.
So I think the head twitch response, I'm unaware of any drugs that are psychedelic, classically psychedelic, that are inactive in the head twitch response.
Yeah.
So I think that's with the cat. So we use these humanized mice now that we've made because there are some differences between the human and the mouse receptor.
These are the mice that have the amino acid difference, right? The
Yeah,
alanine
yeah,
for serine
yeah,
mutation.
yeah. Psychedelics tend to be more potent in them,
Right.
potent and efficacious. We haven't tested a lot of non-psychedelic compounds, so it might be interesting at some point to revalidate those. But I think we're probably like 90%. We can, with 90% confidence, predict whether a compound's going to be psychedelic or not. In terms of therapeutic activity, you have to do the clinical trial,
Mm.
basically.
Yeah, just on the former point there, the head twitch response is a super interesting paradigm in my opinion. It's sort of a curiosity in the 60s when it was developed by Corn and Pickering, and obviously it's become much more important in recent years as people are seeking to therapeutically develop psychedelics. I'm also not aware of any what would be false negative drugs that are psychedelic in
Yeah,
people that don't produce response. But there's certainly a lot of interest in these sort of edge case false positives. Bill Fantagrossi's work claiming that one enantiomer of MDMA but not the other, or the racemate produces a response. Things like Ariadne from Alexander Shulgin's laboratory
mm-hmm.
that do produce a response, but it's a fairly small uptick at a pretty high dose for a drug that is demonstrably not psychedelic in people based on the number of
Right,
people who have taken it,
right.
including some phase 3 studies by Bristol Laboratories many years ago. One of the points I've made to colleagues and in other places is that the head twitch response is highly predictive for very closely related chemical structures with similar pharmacology. There's not really such an easy way to think about how predictive it might be for scaffolds that move very far beyond phenethylamines or
Yeah,
tryptamines. Your lab's proposed some of these from high throughput virtual screening, for example. How do we think about how generalizable the head twitch might be once we move beyond the classical structures that are extremely well characterized at this point?
good question. Just in full disclosure, so I had this DARPA grant, and we have compounds that I guess are property of the Department of Defense and UNC that are novel, novel chemical matter, never seen at the target or reported, that do not, ostensibly are not psychedelic in under any circumstances in mice with multiple assays. But we just never know until you get into you'll find out in phase 1 trials. But use of primates, non-human primates,
Yeah.
that
I,
actually might be the way to go.
Right. Yeah,
Yeah.
I recall a sort of macaque head-to-head study of a putatively non-hallucinogenic compound against LSD. I also thought that was quite interesting, right? So LSD has a pretty distinctive profile in that species in terms of things like its locomotor behavior and some simple tasks. But
And you can
it,
just look at the monkey, basically.
exactly, yeah. I remember thinking for anyone who's done any sort of harm reduction style troop sitting, that it looked a lot like a human on a pretty solid dose of LSD.
Yes.
Yeah, not really moving around a whole lot, looking
No,
around
no.
in sort of wonder at different things. Yeah, it
Yeah.
was pretty clear.
When I was training, so I'm a psychiatrist by training,
Mhm.
and I did my residency at Stanford, and I used to moonlight at the Santa Clara Valley Medical Center, which is, I don't know, 2 miles from Shoreline Amphitheater where the Grateful Dead would play. And I could always tell when the Grateful Dead were playing because the ER would fill up.
Are you a deadhead? Are you a
I
fan?
love the Grateful Dead. Yeah. It's funny. I was never a fan of the Grateful Dead until about literally 20 years ago,
Right.
after Jerry died, basically. And I started listening to them. You have to listen to a lot of Dead to figure out what the hell it is they're trying to do.
Yeah, I have to say I'm not particularly familiar with the Grateful Dead. I have listened to a little bit of their music recorded, but I know countless Grateful Dead tattoos and car decals and other things in Colorado. I think it's because it's a live performance thing, right? Apparently, you have to see them
Yeah, yeah.
live. And
Every concert is every song is different the way they
yeah,
play it. So, yeah,
yeah. It never quite crossed the Pacific to become the same cultural phenomenon in Australia that it is here, and
yeah,
I think that's because of the live
right.
performance angle.
So, right.
Yeah. Yeah. Well, this has been a really, really interesting conversation, Brian. Is there anything else you'd like to discuss before we sort of wrap up here? Really appreciate your time and insights, and looking forward to working with you in future.
The only thing I always say is I like to tell everybody my work is supported by your tax dollars. Um,
Yeah, very important time for that
Very
message, I
important.
think. Yeah.
Yeah. All of my work is supported by the US government. Not only my work, but other people's work should be.
Yep. As I heard recently at an accelerator, we were involved in the One Mind Accelerator, that the US government is essentially an LP in one of the most successful VCs of all time, which is, the National Institutes of Health. And I think a lot of people don't appreciate just how much
Well, they
incredible research, like the work
do
that your
not
lab is doing,
appreciate. Yes.
contributes to life-saving medicines and to other critical technological developments. So very important time for that
And all
message
my
for
work on
sure.
psychedelics, I mean, none of this stuff that I published would be out there, basically.
Yeah, yeah, of
Because
course.
nobody else is going to do it. So.
So yeah, I'm happy to be a US taxpayer for that reason alone. Thanks so much, Brian. It's really great speaking
Yeah.
with you, and looking forward to connecting again soon.